trachea over here, right? And so one of the things that you're going to be responsible for from lab 1 is the positioning of your major organs. And so with this one, we don't have all of the bells and whistles here, but some of the major pieces you should know. We've got the trachea here inside the throat. Down here we've got the lungs in the thoracic cavity. Yes, then we've got the heart inside the pericardial cavity in there, which is a subcompartment of your thoracic cavity. The differentiation between the top cavity and the abdominal cavity underneath is this diaphragm muscle that runs the lungs, and it helps keep that thoracic cavity as a vacuum, which you'll learn in 111 is what actually helps you breathe in and out. Your largest organ outside of your skin is this organ just underneath here, the liver. It's got 3 lobes. To the left, well, to the patient's left, which is always how we're going to do anatomical positioning, right? We've got the stomach here. Behind that stomach, you're going to see a spleen poking out, which we don't see here. And immediately behind both the liver and the stomach, more in the medial area, is your pancreas, if we were to pull these guys out. This gray area here, you can see we've got an ascending colon, transverse colon, descending colon. This is collectively that gray, the large intestine. And then we've got this 25-foot mess going on in between to finish digesting and absorbing your food. That's the small intestine, right? Questions so far? All right, so as we were to pull this guy out, here we have an internal view of the stomach, which is going to flow directly into this first area of the small intestine called the duodenum. That duodenum is loaded with digestive enzymes and really is what finishes up the digestion that your stomach didn't take on. Your stomach really is simply an acid bath to protect you from bacteria that you might eat with your food. And yes, it does do some digestion, but the larger part of your digestion happens in the first parts of your small intestine, your duodenum. Then we get into the absorptive area. This is where you have your columnar epithelia. That's going to be your jejunum. And then we have the finishing area of your small intestine, the last few feet of that, called the ileum, which then goes to the cecum and around the colon that we've already talked about. Questions? As we go a little deeper in here, you can see some kidney back here. That's in the retroperitoneal space. So you should also know about the membranes that surround both of these cavities. We've got the pleural membranes for your thoracic cavity. We've got the peritoneal membranes for the abdominal cavity. If we're to pull that peritoneal wall away, that's when we actually get access to the kidneys and in the back, right? And we have a pancreas here that's just peeking out right in there. It's got more of a mealy, oatmeal kind of texture, and that's typical for glands. These large epithelial cells with lots of ribosomes, lots of Golgi, lots of rough ER, that's going to give them kind of this, well, in real life, an oatmeal under saran wrap type of texture in actual dissections. But here, it's not smooth on the top, but kind of mottled as you go in the back. Your spleen, we still don't see it. It's going to be coming around here on the patient's left side. Down here we have the transverse colon to the descending colon. It's going to hook up with this gray area. That's called the sigmoid colon. And also here we've got the urinary bladder, which will go to the urethra and out for your excretory system. And again, that's something that we focus on digestion and excretion more for 111 than we do here. We're just going through the major landmarks. Here we've got the lobes of the lung. The right side has 3 lobes, the left side, because we've got the heart over here, that gets in the way, it's only got 2 lobes. It's the action of this diaphragm pushing up and down in this vacuum space that allows you to breathe in and out. And you should already be aware from your epithelial lesson that your lung tissue, the alveoli, is made of primarily squamous epithelial cells. These single layer of flat cells that minimize the space that oxygen and carbon dioxide need to cross in order to get from the atmosphere into your blood, right? So there's a purpose to those cells being flat, and it's simply to maximize absorptive potential of the 300 million or more alveoli that you've gotten here that are your air sacs. Now, alveolus in Greek means a bunch of grapes. And so when you were slicing that and looking at that with the epithelial tissues, what we were looking at was functionally the skins of those grapes. 3 million of them in your lungs. Questions here on the general orientation of our buddies? Okay. Now we get into what I'm certain was probably giving most of you fits for the first time this semester, this big long laundry list of anatomical positioning names, which wound up being the bulk, a big deal as far as my labs of what we're examining you on. So we start out here. We've got the frontal portion of your body. The frontal region is the forehead. And it's named appropriately because just under the frontal region, we've got the frontal bone. Just underneath the frontal bone, we've got the frontal lobe of your brain. And so once you know it now, you know it for the rest of the semester for all of the other stuff we're going to be doing to you through the axial skeleton and brain development. The otic area, this is right here around your ears. You're going to learn in the second half of the semester, more of your temporal bone of your skull goes here. For right now, you know it as the otic area. Your orbital area refers to the orbits of your eyes. Yes? And this is just simply knowing what the term means. It's straight memorization. So what you want to do is start your laundry list. And flashcards are a big deal. I'm sure there's a bunch of Kahoots out there, if you like that are already pre-made. You can sign in and do it that way as well. Right here, we've got, well, let's talk about the nasal region. I hope that's not too much of a stretch for everybody, right? The nasal region. And then we've got your oral region for obvious reasons, it's the mouth. Well, the muscles, one of the big muscles that gets you to chew is your buccinator muscle right here. And this is not something we teach you in class, it's something we teach you if you ever take anatomy and physiology for speech, because this is one that matters for how you move your mouth to talk. But as a consequence of the naming of that muscle, the buccinator, we call this the buccal area in your cheek right here. Then we've got your chin, we call that the mental region. Now this one I have no idea why they call it the mental region. It certainly isn't the smart area of your body. However, the mental area is your chin. And we're going to get to you with that when we start talking about your jaw just after the practice with the axial skeleton. We'll see that again. Then anything dealing with the neck here, this is the cervical region. Questions? All right. So then as we continue our inferior dive towards the south end of your body, we keep going. This is your cervical region right there. And then we get into the thoracic region. Now all of these regions of your body, cervical, thoracic, lumbar, these are named according to the vertebrae that are associated with them. And you get down to about C7 right here. The seventh cervical vertebrae is your last vertebrae of your neck. And if you run your finger down your neck, everybody's got this bump right at the base of their neck. That's your seventh cervical vertebrae. The very next vertebrae down, that starts your thoracic vertebrae, which is how we're delineating your thoracic area of your body here. It's functionally your chest. From the bottom of your rib cage, you can feel that xiphoid process, a little sticky thing that sticks down off your breastbone, up to about your C7. That's your thoracic cavity inside. And as a consequence, the thoracic region of your body. Your shoulder here is called the acromion region. And when we get into the scapula, we'll actually see a protrusion of your shoulder blade called the acromial process, which helps make this joint up here, which your humerus plugs into. Yes, so the acromial region is your shoulder. Just underneath that, you've got your armpit. This is the axillary region. I'm pretty sure this is where they got the name for Axe body spray. The axillary region here is your armpit. Then as we move down, here's that diaphragm again, which cuts your entire body in half. It's your skirt steak, if you like skirt steak. The reason why that has a different texture than fillet is because skirt steak is the diaphragm of the cow. And that's how you can get a $22 steak that's the width of your table. Right? It's simply the diaphragm. Why is it this different texture? Because it's a mix of skeletal muscle tissue for the voluntary ability to breathe and smooth muscle tissue for when you're not breathing and your body forces it. Right? And so that's why that's got the different texture than other steaks. Here, we've got in your upper arm, the brachial region. And so this area right here, you already know the names of these muscles because you live in America, right? It's the biceps and the triceps. But the proper names of these muscles is the biceps brachii and the triceps brachii. We get that brachii thing from the fact that it's in the brachial region of your body. Am I making sense? Any questions? All right, so then from there, we can dive deeper into this thoracic cavity. We've got a pleural cavity, which is going to surround your lungs. But in between, intermixed with your right and left pleural cavities, we've got the area around your heart. The around the heart cavity, pericardial. That's what those two terms mean. And if you haven't figured this out yet, anatomy and physiology is a foreign language course with gross pictures. Take it as such. If you learn the language, it's not about memorizing multisyllabic terms anymore. It's about understanding the conversation that's happening around you. And as an intro course, that's one of our main goals with you this next semester, is for you to get into a room full of professionals and simply understand what the hell is happening around you by the words that they're saying. Know the language first, and then know all the other stuff. Is the best way to approach all of this. Yes? All right, so questions on pleural cavity, which holds the lungs, pericardial cavity, which holds onto the heart, inside the thoracic cavity, which is denotated by basically the top here, your collarbone, down to the bottom of your rib cage, where the diaphragm is. Okay. Then we start getting into others. Again, that diaphragm, imagine what I might ask on my practical with this number of diaphragm points to it. Yes? Well, you can talk about some major blood vessels. You've got the superior vena cava, which is bringing blood back from the shoulders and head. We've got the largest blood vessel in your body, the aorta, which comes right off the top of your heart there. That aorta in a blue whale, which is the largest creature that's ever swam or walked the planet, you can take a VW microbus and drive down inside the aorta of that blue whale and never worry about touching the walls of that blood vessel. These blood vessels are huge. And as an artery, they've got the special designation of being surrounded by smooth muscle cells. And I'm sure that your lab instructor pointed that out in the notes during the recitation for lab. And if you didn't, well, that's the difference between arteries and veins. They've got, arteries have smooth muscle around them. Veins are floppy because they do not. And this is more of a 111 type of concept. But those are your major blood vessels. Again, we've got the heart here, left lung and right lung. And this is the sides, remember, we always denote from anatomical position. It's always to the patient's left and to the patient's right as if they're laying down on a gurney. And this way, when we're in the operating room, we don't cut off the wrong leg, right? Which has actually been an issue in the tri-state area over the past few years. We've had a couple people come out of surgery with the wrong limb amputated. So now, these days, we don't even just say, hey, you ready for surgery? You ready to go? Good, let's calm you down and get you in the OR. No, we hand you a Sharpie and you X where they're doing the work. You do that work with the marker now, just to make sure that there's full communication between the patient and their caregivers, yeah? Anatomical position is always to the patient's orientation, left or right, which is why that on your right side is the patient's left lung. All right, so underneath, we've already pointed out, just underneath the diaphragm, your liver and your stomach here. From there, we've got underneath, we've got from the stomach, we go into the small intestine. Your absorptive area of your small intestine, fully 20 plus feet of intestine, is called the jejunum. And with the villi that we looked at in your epithelial tissue slides with that columnar epithelia and this extensive bending around of this jejunum of your small intestine back and forth, your jejunum covers the same surface area as that of an NFL football field. So every potato chip, every burger, every salad you've ever chewed up and swallowed gets spread into a microscopically thin layer across an entire football field's worth of surface area. And what this allows you to do is eat and grab onto those nutrients and put them in the bloodstream as quickly as possible. And this is a theme in your body. It's the same reason why you have 300 million alveoli in your lungs. You increase the surface area, you increase the speed and efficiency of transfer from the environment into you. And make no mistake, that is how you're built. You are a surface area machine. And the excessive length of your small intestine here is just one more example of that. Questions? All right, so from there we've already talked about the ascending, transverse, and descending colon, which is then followed up by a sigmoid colon back deep to the small intestine here. That collectively is your large intestine. The last area of that large intestine here would be the ileum and into the cecum. That cecum will also have an appendix laid after it, we're omnivores. We don't have an all-plant diet anymore, but if you look at the intestines of a rabbit or any other herbivore, that appendix is going to be massive. And what it is a fermentation tank for bacteria. Even rabbits don't have the enzymes to break down grass, but that's all they eat. So how do they do this? They rely on their microbiome, the bacteria that they house in that appendix, to do the digesting for them. Because we've switched to a part animal diet, we no longer have the need to house as many bacteria, which is why our appendix is what we consider a vestigial organ. We don't really use it any longer. The only time we hear about it is when it's getting us into trouble. Well, if we're not using it, why do we still own one? Because generally speaking, it's not killing us before we have kids. And so there's been no evolution away from it. Yes? Questions here? Okay, very good. We're doing all right. And here we have, at the bottom, we talked about the urinary bladder. Here we have a cross-section of the penis with the corpus cavernosa and corpus spongiosum with their urethra coming right through that corpus cavernosa. That will be a Bio 111 concept. It'll be one of the first chapters you get to in your 111 labs. All right, so we can take this abdomen and we can break it up into several areas. And this is really, it's a figment of our imagination, right, these namings, but it helps the conversation along as we're talking about patient care, right? Where is the pain in this patient? Is it in the umbilical region? The umbilical region is right at your belly button, because you probably know that one time we all had an umbilical cord attached there, and that's how mom fed us for the first 9 months of our life. Then, there, belly button is umbilical region. The right and left lumbar regions are directly to the right and left sides of your belly button, and this is named for the lumbar vertebrae that are predominant here just underneath the thoracic vertebrae. Once you get to T12, we switch to L1 through 5, and that's this area right here of your back. We just move that to the front and call that right and left lumbar. Epi means above. Hypo means below or lower. And so when we're talking about epigastric and hypogastric, we consider that umbilical region to be the set point. Above that is epigastric, below that is hypogastric, from the definitions of these root words, epi and hypo. You with me? Now, when we get to your os coxae, that's your hip bones, in the next couple of weeks after the practical, we'll learn that we've got this wing of the pelvis, that's called the iliac crest. And so your iliac regions are basically named after those regions of your pelvis. Whoops. Here and here, right and left iliac, on the right and left here hypogastric. And then we get to the right and left hypochondriac. What does chondro stand for? Cartilage. And so here you should already know that your ribs are made of hyaline cartilage, and that's where this name comes from. You probably also know the word, literally means below the cartilage. So we're talking about this area right here, just beneath your cartilage ribs here. You know hypochondriac is a different meaning, right? What is a hypochondriac? It's somebody who's constantly convinced they're sick when there's nothing wrong with them. This comes from old school medicine before Johns Hopkins was set up in the late 1800s, when doctors would do this kind of thing over their patients. And they'd feel the auras. And basically what they're trying to do is figure out which demons need to be excised. And a medical kit 150 years ago was not a thermometer and a blood pressure cuff. It was a box filled with all sorts of different shaped knives. And if the doctors couldn't figure out what spirit needed to be released, they would say, okay, we need to bleed the hypochondriac area. Because the doctors couldn't figure out what the hell was wrong with you. They'd take a big curved knife and just get it up under your ribs, poke you, and try to bleed you out. The problem with this style of medicine is most of the time half of your patients were dying of the medical treatment, and the other half were dying of the thing we're trying to cure, which doesn't leave us with a whole lot of patients left at the end of the day. Eventually, that definition of doctor can't figure out what was wrong morphed into patients convinced something wrong, and there's nothing wrong with that. And that's interesting when you think about the power dynamics of your doctor-patient relationship, how that type of term would have evolved this way. But that's where we get these terms from. It literally means below the cartilage, here and here, because if you think about that lower rib, and if you haven't yet, you will in the next few weeks, that's mainly cartilage that attaches it to your breastbone. Questions here on this medical region name? Now for your patients, this might be a bit obtuse. And so what we've also done here, because this is where we get a lot of pain complaints coming from, we've simplified it into the right upper, right lower, and left lower quadrant. It depends on your lab instructor which one of these or both they will use. In my classes, you're responsible for both naming systems. Questions here? All right. So then that brings us out to the cell and the microscope. You should also know, for my classes, you should also be able to do the metric system and the definitions of things like kilo and deca and centi and milli, right? All of that we're not going to cover today simply because that's the flashcards. Just go memorize the words, right? I don't need to explain a whole lot besides that. Use your staircase of science to simply move the decimal points around. Don't think too hard, right? We've taught you the staircase of science in that conversions lab simply so you won't have to think. It's not cheating to use the tricks we teach you. All right, so draw it on the back of your practical when you're there if you have any conversion questions. My classes, you will have at least one conversion question station. All right, so when we get to the cell, what we're looking at is specifically the eukaryotic cell. This is the model that we have to test you on. So your best bet is to study this model in particular. So these red guys with the inner workings of membranes, those are the mitochondria. This is where we have ATP production. Once we get to the end of the musculature chapter in your lecture, we'll talk more about how that ATP is actually made. From there, we can also have, what I'm going to do is just move south. This peroxisome is this black circle here. That is a special vacuole which has a lot of things like peroxides and other oxidizing chemicals to help with both immune defense and digestion. In my classes, we can call that a peroxisome. You can also call it a vacuole. Because in truth, nothing inside your cell actually has a color to it, right? So in my classes, that's just as much, might as well be a vacuole. As we move down, we've got this series of flattened sacs, which are going to help fold your proteins in the right way once they've been made by the ribosome. That's called the Golgi apparatus. You might also know this as the shipping and receiving department of the cell, because what that Golgi is going to do is it's going to make vesicles, these little guys here, that are filled with those tightly folded proteins for export from the cell. As we move from there, we've got another circle that's yellow. In this case, it's a lysosome, we're calling it. Lysis means to cut or to slice. Inside that lysosome are digestive enzymes. And so how this is going to work when an organelle gets old, or if this is an immune cell and it just picked up a bacteria by phagocytosis, what we want to do is digest the contents of that vacuole. So we fuse the lysosome with it and expose the contents of these digestive enzymes. They're hydrolytic enzymes, just like your digestive enzymes in your stomach and small intestine, and so they work the same way. We rip the monomers off all of these polymers, and we digest the things chemically. And if it's an immune cell, that's the end of the infection, hopefully. As we keep moving down, we've got this black region here. It really isn't a black region inside a real cell. We just needed some way to delineate for you. That's called the centrosome. That centrosome is a microtubule organizing area. And in particular for us, we're going to get into mitosis very soon. This is where we keep our centrioles, which are the basis of organization for the spindle fibers that will allow anaphase to occur during mitosis. Yes? And we'll get more to that when we get to those particular slides. The centrioles, by the time we get to daughter cells for mitosis, you have one centriole per daughter cell. The last phase of interphase, the last step of interphase into prophase is that one centriole this cell had through interphase will replicate itself, and that will become the two centrioles that we're going to move around in prophase when we get there. Questions? All right, moving further down inside the cell, we've got this big white circle. This is your nucleus, and that nucleus is functionally a chromosome cage. Now, once those chromosomes come out and are condensed in their little suitcases for travel, that's when we call them chromosomes. When they're in interphase in a nucleus, those tightly bound chromosomes, we have to loosen them up because we actually need the physical space to get the enzymes to go in there and read the DNA and make proteins from it and RNAs. And so we can't call them condensed chromosomes when it's in the nucleus and being used. Instead, we call this chromatin. which is a much looser packing of that DNA. And make no mistake, this makes a difference. You've got 6 feet of DNA stacked inside of every single one of your nuclei. Right? That's a whole lot. And so the ability to pack and unpack this stuff for protection during mitosis becomes central to keeping your genetic fidelity generation by generation as you build yourself and repair your tissues. Am I making sense? So when they're out of the nucleus for mitosis, they're called chromosomes. When they're being used for active gene expression inside an intact nucleus, that's called chromatin. All right, so from there, we've got another structure inside these guys called inside the nucleus called the nucleolus. It's a very particular region for RNA creation. All 3 types of your RNA get made here, your tRNAs, your mRNAs, and your ribosomes, which are just ribosomal RNA all tied up into a knot to give us that 3-dimensional structure of the ribosome. All of this stuff, then all of your RNAs get formed inside the nucleus and leave by way of nuclear pores, little holes in the cage that these little messages can sneak through and get out to the cytoplasm. Because RNA doesn't matter unless you can get to a functional ribosome, and those are out inside the cytoplasm. Primarily attached to this greenish blue membrane right here called the rough endoplasmic reticulum. And it's called the rough endoplasmic reticulum because it's studded with ribosomes, kind of like the Michael Jackson 80s jacket with all the metal things on the back of it, right? I might be dating myself, but you know, he's been a thing in the news lately. Well, we keep those ribosomes attached to that rough endoplasmic reticulum close to the nucleus because that RNA is coming from the nucleus, and that's where the RNA needs to get to. RNA is nature's initial self-destruct message. You've got about 45 seconds from release to destruction of that message, so we want to keep those ribosomes convenient. The rough ER is closer to the nucleus. Yes? As we move outwards from the nucleus towards the superficial area of the cell, we've got these dark membranes that are attached. That is the smooth endoplasmic reticulum. In the periphery of the cell, there's not any ribosomes attached to them. Instead, the enzymes for cholesterol synthesis and toxin breakdown are embedded in those enzymes. So a liver cell or hepatocyte would be really, really resplendent in the amount of smooth ER they've got, but a protein-producing cell, like a salivary gland cell or a pancreatic cell making insulin, they're going to be a whole lot heavier in the rough endoplasmic reticulum portion of this stuff. Make sense? Any questions, guys? Joe?
The process of the chromatin leaving the nucleus, now, is that facilitated diffusion, or no? Is that different?
No, facilitated diffusion is when you have a large molecule trying to get into the cell from the outside of the cell through the cell membrane. And what we're going to do with that to facilitate the diffusion is we're going to put an integral membrane protein in the cell membrane, which is going to allow that molecule to come through by way of diffusion, high to low concentration. So we're still doing the same roll downhill thing, passive transport. We're just providing a specialized bridge for it to go on.
I got you.
Make sense?
Yeah.
Others? Yes?
Can you repeat where in the body has a lot of
Places that make a lot of proteins. So we're talking about a lot of your neurons. Many of your glands make protein-based hormones. Any place that makes a lot of proteins, you're going to need a whole lot of rough ER. So just off the top of my head, glands becomes front and center because we've got to fill your bloodstream with these guys to get them working, right? Others? Okay, with a half an hour left, we're not doing that bad. We've got a cytoplasm and a cell membrane that's not just for eukaryotic cells, complex creatures like us. All living things that have a cellular life basis are cell membrane, cytoplasm, DNA, and ribosomes. We're no different, but we specialize in complexity. And that's where we can get the big multicellular version that we are. If you're a bacteria, you don't have any of this garbage. You just keep it to the simple protein production. Bacteria are more successful as a life force than we are as multicellular creatures. They have a different angle on life, though. We go for complexity, which is why we have all these membrane-bound organelles. And that complexity allows us to do what we do as 100 trillion cells apiece, right? But bacteria, they focus on speed. And that speed has allowed them to take over every single environment that we've ever explored on Earth. Because that speed allows a whole lot of fast evolution. And we don't get that benefit. We focus on dealing in the moment. They focus on dealing over generations. But that's the biggest difference between us and bacteria. That cytoplasm is your water basis of life. And the reason why we need water is simply because this is a chemical reaction chamber. It's a chemical mosh pit. And for molecules to interact and create a physiology, they need to smack into each other. So we float them in waters to facilitate the smacking. And we keep them in these tiny little compartments, which basically forces more smacking, which is raising the rate of your metabolism to the point where you can support your life. You with me? All right, so then lastly, what else do we have here? We've talked about the pores, the nuclear pore. I think we're done on this one. Moving along. Then we get to the microscope. We've got a, von Leeuwenhoek built the microscope almost 250 years ago. It's still the most commonly used tool of the biologist today. Because as it comes down to it, if you have an organismal physiological problem, a disease, there will be a chemical basis to that disease in most cases. And as such, we want to get down to the chemical reaction chambers that are causing that metabolism. That's your cells. The microscope is it. You might not know that von Leeuwenhoek was not a biologist. At first, he didn't care about biology. He built his first microscope because he got tired of being ripped off at the shipyard. He was a textile merchant. And so he got tired of being told it was 1,600 thread count, and when he actually sold it to the next guy, it's like 200 thread count. He's getting ripped off. People are taking his shirt. And so, literally. And so now he's got the ability to look at those threads, count them, and say, oh, no, you're not ripping me off. And as a consequence, he got very, very rich. And then he did what in the 17 and early 1800s the rich guys did. They got into explaining the mysteries of the universe. And he focused on the little wee beasties that he could find in his drinking water. Because just like you and I, he found that disgusting and he wanted to learn more about it. And that's how we have the microscope used in biology today. It came from a guy simply trying to stop being ripped off and do his job better. But now we've got all these bells and whistles, and so we call this eyepiece up here with the ocular lens. Okus means eye in Greek, which is where this term comes from. The other set of lenses we've got down here are microscopes. That's the red, yellow, and blue. The red, remember, is not called low power. It's called scanning power, because that's what you're doing with a sucker. You're simply finding your sample with a maximum of wiggle room for field of view and depth of field, or the amount of wiggle room you have for focus, which is why we started red. It's easier to find everything. Then we move to low power, and if need be, high power upon your convenience. In my classes, you're grown. You can make decisions like this on your own. I just ask that you always start on scanning so I don't have to come help you every single time, right? The red is there to give you the maximal wiggle room and still be able to see something under the microscope. After that, we get into our actual magnification. So those are called objective lenses on this revolving nose piece, yes? The arm here is where you're going to carry the sucker, two-hand carry, arm, and base, because we pay for these things and we like to keep your lab fees low. So it's two-handed moving in and out of that cabinet. As we move down the arm, we've got your coarse and fine adjustment knobs. That's moving the stage up and down. And if you know about how that light works, if your lab professor explained this to you, we've got underneath the stage a condenser, a special lens that takes that light coming up from the light source and it focuses it into a cone. An ice cream cone of science, 360 degrees. And the reason why that stage needs to move up and down with the focus knobs is because when your sample is in the peak of that cone, that's when it's in focus. You don't get that cone without the condenser lens up underneath that. A microscope tip for our labs, if you're having trouble finding stuff, basically what you do is you use that condenser knob to roll that lens all the way up to just underneath the stage. Now you've got a pool of light. If your sample's in the pool of light and you could look with your naked eye and see your sample in the pool of light, let's look at that condenser, and it's like an aperture of a camera. We've got that little slide bar in the front of that condenser apparatus. What that's going to do is going to close or open. It's like an f-stop of a camera, if you know anything about photography. It just allows more or less light through. So the two different ways we can adjust our light. You can adjust the actual light with the rolling knob on the base, or you can slide that slider bar and just open and close the diaphragm. So the condenser also comes with that iris diaphragm. and some of your lab instructors have taught you about that as well. Questions? Good question, thank you. Then we get into mitosis. And so we've got our mitosis models, and you should be able to identify the phases of mitosis both in the models and as an arrow on an actual microscope slide. You should be able to do both of these. But here we have an interphase cell. An interphase, at the late interphase, we have now copied our centrioles. We've got two of them. They're going to be on one side of the cell. This is going to be the centrosome of that cell, even though it's not obvious there, right? The nuclear membrane is still intact, and inside we've got loose chromatin during interphase to facilitate gene expression or proteins coming from that DNA. Here's our nucleus, your centrioles, and this red spot here, that's supposed to indicate your nucleolus. As we move into early prophase, what we'll start to see is the chromosomes inside the nucleus start to condense. Instead of being kind of a, well, it starts to look like a bag of worms inside a looser circle as the nuclear membrane starts to dissolve. Your centrioles start migrating to the poles or either side of the cell, and in between them we have these microtubule structures. Those are called spindle fibers. Once we release the hounds from the nucleus, those chromosomes, they'll attach themselves to the spindle fibers and march their way down those spindle fibers towards the centrioles during anaphase. And we'll get there in just a minute. Questions so far? All right, as we move in there, the nucleus, the centriole, your spindle fibers, and these chromosomes now are in sister chromatid form. Remember in interphase, we go through that S phase of the cell cycle where we replicate the DNA. The chromosome 1 and the copy of the chromosome 1, they stick together so far by this central region called a centromere. It's called a dyad. Dia here indicating 2 chromosomes. So they hold themselves together. When anaphase happens, we break the dyad and the sister chromatids go in either direction. 1 per cell. You with me? Questions? Okay. In late prophase, then those dyads are starting to organize themselves with the spindle fibers attaching to the spindle fibers by way of the centromere, which is that central unit of the chromosome. Here are centrioles. They're off to the poles at this point and organizing metaphase. Meta means middle, so what we've got is these dyads now all lining up towards the middle, equidistant generally from chromosomes to centriole on either side. We call this the equatorial plate, the area between the centrioles that the chromosomes lining up on. Now, not only do we need to attach these chromosomes to the spindle fibers, but we've also got to make the dance floor bigger so we can do all these moves. So we've also got these aster rays, which go in the opposite direction towards the outside of those cells. And these are still microtubule tracts. They're legitimate cytoskeletal elements. What they're doing is they're pushing the cell membrane out and giving us the maximum amount of space in the middle of the cell to make all these chromosomal movements happen. If we don't have enough space, the chromosomes' arms start to wrap on each other and they start to break. And that's going to be really bad for the daughter cell if they don't have whole areas of chromosome coming into them, right? And so this here is the same reason why the chromosomes condense before they're allowed out of the nucleus. It's to avoid chromosomal damage and chromosomal hanging up on each other, which would then rip your DNA right in half and likely cause the death of the daughter cells. Make sense? Questions? Okay. Then we get into anaphase. You know why they call it anatomy? What the root of that term anatomy is? What ana means? It means to slice up. How did we figure out your parts inside? We slice things up. That's where the term anatomy comes from. It literally means to cut up. And so here when we're talking about anaphase, we're using that same root word, ana, to mean the same thing. We're cutting up. And what are we cutting? We're taking these dyads, these pairs of copied chromosomes, splitting them down the middle and sending one copy that way and another copy that way. We're cutting those chromosomes up, one for you, one for me, one for you. All 23 pair move this way. And so anaphase, what's happening is the centromere has a motor on it called a kinetochore. Kinetic, meaning movement, right? That kinetochore literally chews its way down the spindle fibers, spitting out that railway track in pieces behind it until they get down to the centrioles, which are the whole organizing region of this. Once we're down here by the centrioles, now all of those chromosomes start to recondense. We start to get a nuclear membrane back around them. That's called telophase. So here, now we are no longer dyads in anaphase. We've cut them up. That's anaphase. We call them now chromatids. And this is the nitpicky BS language stuff that your instructor might throw at you. I likely will as well, because it's a good way for us to figure out who's doing the work and who's phoning it in. Right? And that's really one of the things we try to do with you in this course. It's kind of a weed out course. We're not going to deny it. And this is the kind of stuff that helps us figure out the workers from the non-workers. Yeah? Aside from being legitimate vocabulary terms. All right. Centrioles, spindle fibers, and the asterades, which are making our flora a little bit bigger to dance on. Late anaphase now, we can start to see the beginnings of cytokinesis. Understand and remember, mitosis is about anything that's happening of the nucleus and chromosomes. It has nothing to do with the cytoplasm. Cytokinesis is all the cytoplasm and organelles split up. It's a different process superimposed on top of this nuclear process. So sometimes cytokinesis starts in late anaphase, sometimes cytokinesis starts in early telophase. But when you see this cleavage furrow start to form, that's when you've got cytokinesis happening. It's a separate thing from mitosis. Yes? All right. From there, we've got the exact same pieces we've looked at before, so I'm not going to insult your intelligence by going over them again. The only thing we have to add here is that cleavage furrow to indicate that cell stuff is starting to split as well. Then telophase. Telo comes from a root word teleo, which means the end point, the end game, where we're supposed to be. And if you think about mitosis, what we're supposed to be doing is getting our chromatids now into a new nucleus. And so that's the end point, is to put these chromosomes in the new nucleus. That's where the term comes from, telophase. We're going to see these chromatids start to condense again to form chromatin, looser expressive DNA, and we're going to see that nuclear membrane form around both sides. From there, we've talked about all of that. And then we've got our two daughter cells. That cleavage furrow continues to pinch in until this side meets this side, and we finish up the membrane. Now we've got two daughter cells. The nucleus is fully reformed. The chromosomes inside have now relaxed so we can get the protein expression machinery in there. Now they're back to chromatin. In slide form, what we've got here is a interphase cell. Cytoplasm with a cell membrane on the outside. If you look at that nucleus, it's very flat in its staining pattern. It usually doesn't have a whole lot of modeling or freckling or variation in the color tone. And that's because chromatin is loosey-goosey all throughout that nucleus. It's like the chromosomes gone on their beanbag chairs and like, yeah, chill out. Well, that's going to lead to this type of staining pattern for most of our slides that we have. And when we get into prophase, take a look at this sucker. The nucleus is quite a bit different looking. Now it's got this modeling or freckling, or I call it a bag of worms type thing, where it's very clear dark and light areas. The other thing that's going to be different is that the ring of that nucleus becomes very fuzzy. And this is because that nuclear envelope, that's going away. And so we're going to see that as a fuzzier border around the nucleus. Couple that with this light and dark type of staining, that's the chromosomes now condensing into their mitotic form. Prophase for sure. No nuclear envelope or a very minimal presence. And now these dyads start to float around inside the cytoplasm where they'll attach to the spindle fibers. From there then, we go from prophase to metaphase. We've got our centrioles on either side here. We've got the equatorial plate, and you can see a dark staining. My pointer's not working, so this dark staining here, metaphase. Probably the easiest one to recognize. Then from there, we have A equals away, this Anna cutting up the the dyads into the sister chromatids, and we can start to see we've got these guys splitting apart. You also in this slide see a cleavage furrow starting to form, so this must be a late anaphase slide. How we ask these questions, we put the slide inside the microscope. Most of the microscopes have that shadow arrow in there. We roll that arrow, we point to a cell, and say, what phase of mitosis is it? And then some follow-up question to go along with it. At least how it happens in my class. All right, and then we get the telophase. You can see a very distinct cleavage furrow. You can see that that cell membrane between the two is pretty well finished, dividing these two cells into the two daughter cells. We can see that the chromosomes are very tight now on either side, and they're starting to get more of a circular appearance as the nuclear membrane starts to reform around those chromatids. And then we go from here to daughter cells, in which basically the only difference is two interphase cells sitting right next door to each other.
So if you wrote telophase and daughter cells, is that wrong? Because it's just like...
Telophase, okay, so you can say late telophase and possibly get away with daughter cells?
Because I wrote both.
You wrote
I
both?
already took it.
Okay.
I still wanted to come. But like, I wrote both because I was like, I know this is definitely daughter cells because they were like the two things. But then I was like, it's asking what phase. So then I'm like, well then it would have to be that.
If they're daughter cells and fully separated, we're really looking in an interphase here, so you might get pegged on the details of it.
It was like still connected.
But if it's still connected, then you can probably talk about being telophase. But if you're really being nitpicky, if your professor's really nitpicky, interphase and daughter cells are the equivalent, not
So then it's
telophase
gonna
and
start
daughter
looking
cells.
at it.
Others? All right. Then we get into our tissues, and we've got 11 minutes left, so I'll try to make this as straightforward as possible. Simple versus stratified. Simple is a single layer of epithelial cells, and remember what these cells are. They are many cells, single file lines, making huge walls and fences, zippered tightly together, so there's a very minimum of ability to wiggle through the alleyways between these cells. This is lung tissue. This is your alveolus, or your bunch of grapes. And if we were to take a bunch of grapes and slice it all Fruit Ninja style, what we're looking at is the skin of those grapes. This is air. We've got blood vessels and capillaries and connective tissue around in the wings to accept that oxygen and to give up the carbon dioxide across a very minimal biological layer here. That's why it's squamous or flat, in order to maximize the ability for gas exchange here. Questions? From there, we've got your simple columnar epithelia. Two major places you can look at this. We showed you the trachea with the ciliary elevator associated with it. You could also talk about your jejunum or your absorptive area of your small intestine. That's what this is. Columnar epithelia, simple because it's a single layer. Columnar because they look like columns. Why do we need this extra space? Why are they not flat? Because there's a lot of regulatory and checkpoint proteins that your nutrients go through before we give you access to the blood. We're making sure that your food didn't poison you. And then from here, this blood, the very first place it goes after your intestine is your liver to give us one more shot at detoxification before we expose your heart, your brain, your lungs to that kind of stuff, right? Inside that columnar epithelia, what we've got are these bluish guys. Those are called goblet cells. These make the mucus that's going to help lubricate the flow of your foodstuffs as they move through these pipes. This is the apical end of the cells. In any epithelial tissue, you're going to need a basement membrane, and that is superficial to the lumen. The lumen is just the center of the pipe where the stuff flows through, right? And so here we have your absorptive surface area. You've got over 20 feet of pipe with this level of organization there so we can absorb all your nutrients. Questions on simple columnar epithelium. For your practicals, you will likely need to identify both the style of epithelia, squamous, cuboidal, or columnar, and also is it simple or stratified. Stratified simply means layers, so if you see more than one layer, it's stratified. It's very simple, that's it. If it's a single file line like this, it's simple. Goblet cells are embedded in here to give us the mucus we need for the overall physiology of the organ. Here we have pseudostratified epithelia. In my classes, we did not cover this. Oh, I did. Oh, simple columnar, simple cuboidal. Thank you very much. A simple cuboidal, these guys look like that. They look like cubes. And so we've got some physiology happening in here. And what we're looking at here, this is your kidney tubules. So this is a pipe that's sticking out from the screen and then diving back into the screen. And we go into renal physiology in 111 as well. This is your excretory system. This is what's making urine for you, getting rid of a lot of the poisons from your blood. And so this ring here, your basement membrane, is going to be superficial to the lumen. Your apical end points in towards the lumen, and the function here is absorption and secretion, particularly in these kidney level cells. The rest of this out here, these are all each individual circle that has a large lumen. Those are your kidney tubules, and they're made up by those simple cuboidal cells. Stratified squamous epithelia is what you see on the insides of your mouth. If you did the whole cheek swab to look at your own cells, the reason why it just took a bit of cotton to rip those cells off is that they're designed to fall off. In this way, what happens is, as that Dorito that you didn't quite chew all the way starts pushing up against the sides of your pipes, it's going to scrape off these cells up top, but leave many layers of protection as reinforcements behind. And so your entire digestive tract is loaded with a stratified squamous epithelial layer because those guys fall off and lubricate the movement of those foods as they move through. The other place you have a stratified squamous epithelia is on your skin, right? Where the top 20 or 30 layers of those suckers are dead, so you can avoid pathogen attack for the most part. Bacteria can't eat keratin, and viruses can't take over dead cells. So we surround you with dead cells to make sure. Transitional epithelium. Did your instructors cover this one? This used to be in the curriculum, it is not, so I'm just going to keep moving. And then we get into connective tissue. Blood is going to be one that we cover, although we didn't force you to draw it. You probably should have looked at it. Primarily red blood cells, or erythrocytes. Erythro just means red. Thrombocytes are your platelets for clotting. Leukocytes, leuko means white. They stain purple the way we stain them, but if you're actually looking at the actual real blood, they're whitish, so we call them white blood cells. That's for your immunity. You'll get into what the different style of nuclei in your white blood cells mean next semester when you hit 111. Because each type of white blood cell does a different specific job. Some are for worms, some are for viruses, some are for bacteria. We have bone, which is connective tissue. We organize these osteons in these lamellar layers, laminate layers, again, just meaning layer. If you've ever laminated anything, it's just putting some paper in between plastic. For me, I got kids, so I can have them mark up the thing. I can just use some Windex to clean it off, right? Well, we're using that in the same way, laminate layers. In between here, what we've got are these dark circles. These dark guys, those are called lacunae, or little lakes, and inside those little lakes is where your osteocytes live, your bone-maintaining cells. At the very center of each osteon is going to be a central canal which contains an artery, a vein, and a nerve. The osteocytes talk to each other very actively. Your bone is an active, living, always redefining itself tissue, and it's by way of these conversations the osteocytes have through the canaliculi, tiny canals that attach each little lake to each other. And in this way, all the osteocytes can set up their own multi-point internet system and always talk to each other. You change your shoes, by the end of the day, your bones are starting to rebuild themselves to deal with the new stresses of your life. We call that central canal in older textbooks Haversian canals, so you should be well versed in both definitions. They're both just the blood vessel and nerve hole that runs down the center of each osteon. We've got cartilage, mainly for your ribs and the ends of your long bones, is hyaline cartilage. And again, connective tissue is a few cells and a lot of extracellular matrix. In this case, the extracellular matrix is chondroitin sulfate and collagen, which is going to give cartilage that bouncy feel. If you like chicken legs, that little bouncy tab at the top of a chicken leg that you try to chew but your teeth bounce off of it, that's hyaline cartilage. You've got that at the end of your long bones as well. Cartilage cells are called chondrocytes from the Greek chondro, which we're going to take to mean in this course cartilage. Elastic cartilage, we're not going to go over just for time. Fibrocartilage, we don't have time for. I've got 3 minutes left before I need to get to my own class. Areolar connective tissue is made primarily of fibroblasts. Fibers we take to mean ropes here, and primarily the main rope is collagen. We can also have things like reticular fibers and elastic fibers that go in here as well. Your white fibrous connective tissue is tendons and ligaments, and you can see we've got a collagen matrix with these very elongated, wavy nuclei that go through. Adipocytes are your fat tissue, primarily around your eyeballs, around your kidneys, and underneath your skin and your hypodermis. All the white space here is the actual lipid. Your cell, the nucleus and the cytoplasm, is just relegated to these tiny little corners. And the extracellular matrix of your adipose tissue or fat tissue is areolar connective tissue. We tie all these guys together with collagen and elastin fibers. We talked about the bone. I don't have time to go over the model, but this is the same thing as our bone histograph that we just gave you. And then your muscle tissue types with the remaining 3 minutes. Skeletal muscle is voluntary muscle. As such, this is what's getting you up and down the stairs. This is what's pulling the kitty litter out of the trunk. We need power moves here. And so with skeletal muscle, what we've got is several cells, sometimes dozens of cells that have fused together in your development, leaving a multinucleate rod that can then have these striations, which are all reflective of the actin and myosin myofibrils inside, which become this contractile fibers of your sarcomeres. And you're going to get to that in nauseating detail in your lecture if you haven't already. Skeletal muscle tissue, these cells can be up to 1/3 of a meter long. They run the length of your muscle. And what we're doing is we're tying all these tiny molecular little contractions together into one large, huge power thrust. This is 1/3 of your body mass because you're an animal and getting around is kind of important to us. Yes, it's all your skeletal muscle is voluntary, which means every single rod that you've got, skeletal muscle fiber, is connected to your nervous system directly. Each has its own individual say right to your brain. Cardiac muscle is a little bit different. Cardiac muscle can control their own contractions. And so what we've got to do with cardiac muscle is tell the heart muscle once at your right sinoatrial node. That's what your brain talks to. And then from there, your cardiac muscle cells play this game of cellular telephone. Did you play this when you were in kindergarten? Where you just say, okay, the teacher says, and then that kid tells that kid, then that kid, and then that kid, and then that kid gets back to the teacher, and it's always different, and everybody laughs, right? Well, that's how your cardiac muscle cells work. And so what we need to do here is we need to facilitate the communication cell by cell. We do that by way of these perpendicular dark lines. And those perpendicular dark lines here, 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 here. That's the easiest way to tell the difference between skeletal and cardiac muscle tissue. Those are the intercalating discs. Those discs provide a direct cytoplasm to cytoplasm connection between these cells. So this cell can talk very quickly to the next cell. And we can contract those cells one by one down the outside of their heart and up the middle and give yourself a proper heartbeat. We'll get more into that when you talk about the heart in 111. But that's skeletal muscle, cardiac muscle tissue branchy with intercalated discs is what you're looking for there. Smooth muscle tissue works as tiny little individuals. You don't get striations with this guy because the myofibrils are just haphazardly organized inside of there. And they work as individual cells. That's why in lab we gave you that teased muscle slide cell to give you. To draw, just to make the point that smooth muscle cells are not collections of easily talking cells. They're all individual cells, each giving a tiny squeeze. So we wrap your esophagus, we wrap your ureters, we wrap your arteries. Any place we need to push fluids through a lumen, we wrap those tubes with smooth muscle so they can progressively contract, pushing that stuff through by way of this mechanism called peristalsis, which I'm sure you've heard of. All right, and lastly, guys, the skin. Where we've got the epidermis on top, that's the purple area. We start with the stratum basale, we get up to the stratum corneum. You learned about that in lecture. We've got your dermis down here, which is largely connective. That's areolar connective tissue, primarily collagen, which is why you can move your skin back and forth. That's the dermis underneath of connective tissue, giving you that level of flexibility. As you age, collagen production goes down. This is a known thing that happens to everybody, which is why everything sags as you get older. It's just about your dermis not tying your skin to your muscles as well any longer. Truly. I used to be a chemist at Estee Lauder. One of the big things they're doing over there is trying to replace the collagen in your dermis to reduce the appearance of aging. Right? They're not reducing the aging, they're still working on that. Because this is all connective tissue, we can start embedding things. We've got the hair follicle with the sebaceous gland that makes hair oil, antibacterial, and it also keeps the hard keratin of your hair nice and supple to avoid damage. Then we've got your sweat glands, sudoriferous glands, two types. Eccrine sweat gland for thermoregulatory sweat. That's your human survival adaptation. We are the naked ape because we have more thermoregulatory sweat glands than any other animal. That's why we can win every couple of years in the horse versus person marathon that goes over out in some bar in the UK over there. Last year, humans won versus a horse over 25 miles. Why? Because we can keep running. The horse has to sit down and cool off or die. And that's why we don't have hair across the rest of our bodies, to help that thing work. The other thing we can talk about here is your erector pili muscle, which is going to take whatever hair we have left and cause goosebumps, which is then to bring that hair up. What this is it used to be a mechanism to trap air underneath our hair, which is a nice insulator. Now that we're the naked ape, we don't have that anymore. We just get goosebumps. So if you get scared or if you get cold, you're going to see that erector pili muscle working well. Then and lastly, we've got the hypodermis, which really isn't part of the skin at all, but a lower level of the fascia, but we call it the beneath the dermis. This is primarily adipose tissue, wrapping yourself in a fat jacket to control your body temperature. And that's labs 1 through 7, guys. I hope it helped. Good luck on your practicals.